Hello everyone and welcome back. As we have started the chapter Biological Molecules, today we shall continue our journey on the road of building blocks of life, and we shall discuss DNA today, which are essential bricks of our biological system. My dear students, if proteins are the machines, the DNA is the master blueprint which tells us the cell exactly how to build them. DNA is a massive polymer, and just like starch is made of sugar and proteins are made of amino acids, DNA is built from repeating units called nucleotides. Think of a nucleotide as a three-part chemical brick. Every single one contains a 5-carbon sugar. You can see this 5-carbon sugar, okay, specifically deoxyribose. That is where the D in DNA comes from. Number two is a phosphate group. This is the glue which links the bricks together, and an organic base. Okay, this is the most important part. This is the only variable part of the brick, and it is what actually carries the genetic code. When these nucleotides join up, the sugar of one molecule snaps onto the phosphate of the next, and this creates what we call the sugar phosphate backbone. It is incredibly strong and stable, acting like a protective outer casing for the delicate base tucked away inside. Okay, you can see this is the DNA molecule, okay, and this is the RNA molecule, and here this is the 5-carbon sugar, okay. There is an organic base and there is a phosphate group, and these are the linking nucleotides. The nucleotides are being linked to form a chain. This is the final piece of the chemical puzzle. So far, we have learned about carbon, hydrogen, oxygen, nitrogen, and sulfur. Now, if you see the element that is a phosphorus in a molecular diagram, this one is here, okay, phosphate group, this is a phosphorus in a molecular diagram, this is in a molecular diagram. If you see phosphorus in a molecular diagram, you are looking at a nucleic acid. Remember now this hierarchy for your exam. Carbohydrates and lipids, they contain carbon, hydrogen, and oxygen. Proteins, they also contain carbon, hydrogen, and oxygen, and in addition to these three, they also have nitrogen or may have sulfur. And in DNA, carbon, hydrogen, and oxygen are present. Nitrogen is also there. But how you can distinguish between protein and DNA? If you see a molecular diagram, in protein there is no phosphorus, but in DNA there is a phosphorus. Okay, there is P, that is phosphorus. So carbon, hydrogen, and oxygen are basic elements in each uh, biological molecule. Okay, but proteins have nitrogen and sulfur, and DNA have phosphorus in addition to carbon, hydrogen, oxygen, and nitrogen. So spot the P. If you spot the P, then you have spotted the DNA. As we have seen the backbone of DNA, but now let's look at the actual ink in the blueprint. While the sugar and phosphate stay the same in every nucleotide, the organic base that changes. Okay, organic base changes. There are 4 different bases, and we usually just call them by the initials like A, T, C, and G. And if you want to know the complete name, and you must know the complete name of these alphabets, A is for adenine, C is for cytosine, G is for guanine, and T is for thymine. Remember these words: adenine, thymine, cytosine, and guanine. Think of these as a 4-letter alphabet. The meaning of your DNA is not the chemicals themselves, but in their sequence. For example, for example, the sequence ATGC might mean something completely different from if we repeat this in this sequence CGTA. ATGC might mean something completely different than CGTA. So inside your cells, the DNA is read in groups of 3. and every three base is called a codon, which acts as a code for one specific amino acids. Here, this is codon. A sequence of three bases usually codes for one specific amino acids, that is a codon. This is the missing link. The order of bases in your DNA directly determines the order of amino acids in your proteins, which, as we have learned earlier, it determines the protein's 3D shape and its ability to do its job. So, as an example for you, this is a very common long answer question. How does DNA actually control the cell? To get good marks, even high marks, you must follow the logical chain. DNA has a sequence of bases. This sequence acts as a code, and the code determines the order of amino acids. I repeat it. DNA has a sequence of bases, and this sequence acts as a code, and the code determines the order of amino acids. The amino acids fold into a specific protein like an enzyme. That enzyme then controls the chemical reaction in the cell. So do not forget the nitrogen check here. These are nitrogenous bases, which means they are packed with nitrogen. This is why organisms need a constant supply of nitrogen to build both their proteins and their DNA. DNA nucleotides do not just float around randomly. They join together to form a single incredibly stable strand. This is the foundation of the DNA molecule. So look at the connection here. The phosphate group of one nucleotide bonds to the deoxyribose sugar of the next. This repeating pattern is what we call the sugar-phosphate backbone. Okay, sugar-phosphate backbone. Think of a zipper. The backbone is the fabric tape on the side, and the organic bases are the teeth sticking out toward the middle. Because the backbone is on the outside, it acts like a protective shield, keeping the delicate genetic information, and the bases are tucked safely away on the inside. You can see that in this picture. This is very clear here. Okay, as I told you in this slide, that this backbone, like this is like a zipper. Okay, the backbone is the fabric tape on the side. This is the fabric tape. Okay, this below and this yellow color, and the organic bases. These are the organic bases, like a ladder. These organic bases are the teeth sticking out toward the middle, and this is the sugar phosphate backbone. As a takeaway tip for your exam, you need to understand the difference between the strong and weak parts of DNA. The bonds in the backbone are covalent. Okay, the bonds in this backbone, sugar phosphate backbone, these are covalent. These are very strong. They act like permanent glue to make sure your genetic code does not just snap into pieces. Okay, in the next slide we shall see the hydrogen bonds in the middle, those which are weak, which is vital because it allows the DNA to unzip when the cell needs to read the instructions. So let's move on to our next slide. Now let's put it all together to see the most famous shape in biology, the double helix. DNA is not just a single chain. It is actually made of two strands which wrap around each other like a twisted rope ladder. These are two strands. These two strands are held together in the middle by weak hydrogen bonds between the bases. But the bases do not just pick any partner. Okay, these bases do not just pick any partner randomly. They follow a very strict, predictable rule called complementary base pairing. Okay, and remember this base pairing. This it will be very helpful for your future studies. Adenine, this is the adenine. Adenine always pairs with thymine. Okay, adenine always pairs with thymine. And the cytosine, it always pairs with guanine. This is a universal truth. Okay, and think of it as a ladder. The side ropes are your sugar phosphate backbones, and the rings or steps you would stand on are the base pairs. And finally, grab that ladder and give it a twist. That spiral shape is the double helix. My dear students, examiners love to give you a math puzzle based on this. Because adenine always pairs with thymine, their percentages must be equal. For example, if a sample is 20% adenine, it must also be 20% thymine. It means adenine and thymine should be equal in number, in quantity. You can, for your understanding, okay? That accounts for 40%. Mean adenine and thymine collectively counts for 40%, 20% of each. So the remaining 60% is split equally between cytosine and guanine, meaning 30% cytosine and 30% guanine. So for those students who are looking for A grade, adenine and thymine are held by two hydrogen bonds. Remember it. Adenine and thymine means these two, adenine and thymine, these are held by two hydrogen bonds, while cytosine and guanine, they are held by three bonds. This means DNA with a lot of cytosine and guanine pairs is actually slightly harder to pull apart. Why? Because there are three bonds. Now, as we have reached the end of our journey through the blueprint of life, this table is your final at a glance check. If you can memorize these rows, you are 100% ready to move into next lecture. Okay, so remember, the monomer is the nucleotide. It is made of that deoxyribose sugar and a phosphate group, which form the strong covalent sides of our ladder. The information itself is held in the base pairs, adenine with thymine and cytosine with guanine. I have written that here. Okay, adenine with thymine and cytosine with guanine, which are held together by those zip-like hydrogen bonds. And of course, the final 3D masterpiece is the double helix. Let's avoid the most common error in your exam. Okay, confusing which confuse you. Do not get confused about monomers. Okay, if you're talking about proteins, the building blocks is an amino acid. But if you're talking about DNA, the building block is a nucleotide. So do not swap them. One thing you should remember, that one difference is DNA has a phosphorus, protein does not have. Secondly, the building block of a protein is amino acid, but building block of a DNA is a nucleotide. And finally, consider a universal rule. DNA is the exact same molecule, whether it is in a bacterium, a blade of grass, or you. The chemical bricks are identical. The only thing that changes is the sequence of those four bases. Which are four bases? Adenine, thymine, cytosine, and guanine. This is the foundation of modern medicine. It is the reason we can take a human gene and paste it into a bacteria to produce life-saving insulin. Now, I hope you have mastered the molecules of life, but this is not the last one. No, we should know that who have contributed to this theory, who have worked so enormously to bring us this knowledge of DNA. So we shall discuss DNA detective story. Okay, we often talk about the double helix as if it were common knowledge, but the journey to find it was one of the greatest scientific detective stories ever told. It was not solved by one person in a lab, it was a puzzle pieced together using clues from chemistry, physics, 3D modeling. Our first detective was Erwin Chargaff, who was a chemist. He noticed a pattern in every DNA sample he tested. The amount of adenine always matched the thymine, and the cytosine always matched the guanine. We now call these as Chargaff's rules. Okay, Chargaff rules. At the time, he did not know why, but he knew it meant these bases must be connected as partners. Adenine, thymine, cytosine, guanine, they are partners. Then the physicists stepped in. Rosalind Franklin and Wilkins. They used a technique called X-ray crystallography. They shone X-rays through crystallized DNA to see how the rays scattered. This led to the creation of the most famous image in biology. We call that image as photo 51. In next slide, I shall show you that photo 51, okay? So when Franklin saw that distinct X pattern, she realized something profound. DNA was not a straight line. She came to know that DNA was not a straight line, it was a helix. Her measurements also revealed exactly how wide the molecule was. These two clues, the chemical ratios of the bases, okay, and the physical shape, were the final pieces which allowed the structure of life to be unlocked. The final piece of the puzzle fell into place in a lab in Cambridge. While Sherlock was busy with chemicals and Franklin was capturing X-rays, James Watson and Francis Crick were acting as the architects. They did not actually perform lab experiments. Instead, they took the data from everyone else and tried to build a physical 3D model using metal plates and rods. For a long time, they could not get the pieces to fit, but then they had their breakthrough. They realized that if they followed Shagaf's rules, pairing A with T and C with G, the ladder would have a perfectly constant width. This matched Franklin's photo 51. This is photo 51, okay? Uh, this one is called as photo 51. You can see this X-shaped figure here. So the arrangement of cytosine of adenine with the thiamine and cytosine with guanine, this matched the Franklin's photo 51 measurements exactly. This geometry worked, the chemistry worked, and the double helix was born. So as an exam tip for you, modern examiners are very interested in the ethics of science. You may be asked about the importance of collaboration. It is important to note that Rosalind Franklin's contribution was largely overlooked during her lifetime. We shall discuss it in the next slide, okay? That was overlooked even though Watson and Crick could not have built their model without her for photo 51. So if you see a grainy black and white X, grainy white and black X made of dots on your exam paper, they recognize it immediately. This is photo 51. This is photo 51. It is arguably the most important image in the history of biology because it proved the spiral shape of our genetic code. Now, it is a common myth that Watson and Crick just woke up one day and knew the shape of DNA. In reality, they built several failed models, which were completely wrong. To find the right structure, they had to solve two major Goldilocks problems. First of all, they made the outside in mistake. Okay, outside in mistake. Their early designs put the backbone on the inside and the bases on the outside. But chemistry said no. The bases are hydrophobic. They fear water, while the phosphate backbone is hydrophilic. It loves water. Since the cell is mostly water, the bases had to be tucked safely away on the inside, shielded by the backbone. Then came the size problem. Size problem. Not all bases are created equal. For example, giants, which are also called as purines, means adenine and guanine. Okay, now you should also know a new term: adenine and guanine, those are purines, and cytosine, thymine, those are pyrimidines. These are small bases. Large bases are adenine, guanine, and small bases are cytosine and thymine. So adenine and guanine are large with a double ring structure, but cytosine and thymine, those are smaller with only a single ring structure. So if you paired a large purine with another large purine, the DNA would bulge out. So if you paired two small pyrimidines, the chain would be too narrow. The ladder would be bumpy. Means one step would be large, would be bigger, and the second step would be smaller. So to keep the width of a DNA perfectly consistent, exactly as Rosalind Franklin's data showed, they realized you must always pair a base with a small base, means you should always pair purine with a pyrimidine. It had to be just right. This is the physical reason why adenine only fits with thymine and cytosine only fits with guanine. Like this one, you can see that size problem here in this picture, and if you understand this one, you can exactly understand the helical structure or double helix structure of the DNA. So why must A pair with T? Means why must adenine pair with thymine? Why can't we have an adenine guanine pair? It all comes down to the laws of geometry and the Goldilocks fit. If we try to pair two large bases together like an adenine and an adenine, the DNA chain would bulge outward. This is a very irregular structure, large plus large pairing, and this is small plus small pairing. Okay, so it will be too wide if we pair large with large. On the flip side, if we pair two small bases like cytosine and cytosine, the chain would be too narrow to bridge the gap between the two backbones. The ladder would be impossible to climb. The only solution that works is pairing large base with a small base, and this creates a perfectly uniform width of exactly 2 nanometers. This one is 2 nanometer. This was not a guess. It matched Rosalind Franklin's X-ray measurements to the decimal point. So, if you are asked why adenine only pairs with thymine, you need to provide two specific reasons. First reason is size. A large purine must pair with a small pyrimidine to keep the DNA width consistent. And second reason is hydrogen bonding. They have to be chemically sticky for each other. Adenine and thymine form two hydrogen bonds, while cytosine and guanine they form three. They are like pieces of a puzzle that only click when they find their specific partner. So remember, Watson and Crick did not discover this under a microscope. They proved it through a logic by showing that this was the only model that did not break the laws of physics. In 1953, the world finally saw the finished blueprint of DNA. The Watson and Crick model revealed three critical features that define every living thing on Earth today. First is the double helix. We are talking about two separate strands of polynucleotides twisted around each other in a right-handed spiral. This shape is a mathematical masterpiece. It is incredibly stable and it allows a DNA molecule which is about 2 meters long if you stretched it out. Okay, so to fold and coil tightly enough to fit inside a microscopic cell nucleus, it is the ultimate space saver. Next, look at the anti-parallel strands. This is one of the coolest parts of DNA architecture. The two sugar phosphate backbones actually run in opposite. Like this as a theoretically it has been shown here. Think of it like a two-way street. One set goes up, running from 5 prime to carbon prime. Okay, from 5 to 3 prime, while the other side goes down, means from 3 prime to 5 prime. They are parallel, but they are oriented in reverse. This anti-parallel setup is essential because it allows the nitrogenous bases to line up perfectly in the center to form those hydrogen bonds. And without this flip, the puzzle pieces would not click together. If you came to know, if this came to into your notice, that I gave a word that is prime. Okay, so whenever you study this thing is 3 and there is a symbol with 3 and 5 and there is a symbol with this 5, and you are talking about DNA nucleotide or double helical structure, double helix of this DNA model, then always you can see that this is 3 prime and this is 5 prime. Okay, finally let's look at the rungs of our ladder. These are the base pairs, adenine with thymine and cytosine with guanine. These rungs are held together by hydrogen bonds. Now in chemistry, we often think of hydrogen bonds as weak, but in DNA, they are the perfect strength. They are strong enough to keep the two strands locked together in a stable helix, but weak enough to unzip like a zipper when the cell needs to replicate or read the genetic code. So my dear students, you must understand the 5 prime to 3 prime rule. I am again saying this word, 5 prime to 3 prime rule. DNA is not just a random pile of letters. It has a very specific direction. The enzymes which build your DNA, like DNA polymerase, are very picky. They can only add new nucleotides to the 3 prime end. Okay? They can only add new nucleotides to the 3 prime end. This means DNA is always read and built in one direction. And here is a top tier tip for your structured questions. Count the bonds. Okay, adenine and thymine have 2 hydrogen bonds. Cytosine and guanine have 3 hydrogen bonds. This actually has a biological sequence. If a piece of DNA has a high cytosine guanine content, it is much more stable and harder to melt or unzip because of those extra bonds. It is like having extra stitches in a seam. It is just plain stronger. Okay. This in this picture you can see hydrogen bond, that is double bond here between adenine and thymine, and 3 hydrogen bonds between cytosine and guanine. Okay, it is clear from this picture. While Watson, Crick and Wickens were awarded the Nobel Prize in 1962, the story of the double helix is incomplete without acknowledging the invisible contributor, Rosalind Franklin. You might wonder why she did not share that Nobel Prize. The answer is just very sad. The Nobel Prize is not awarded posthumously, means it is not awarded after death. Franklin passed away in 1958 at the age of just 37, just 4 years before the prize was given. However, her work was a silver bullet. Photo 51 was not just a pretty picture, it was the definitive piece of evidence that provided the exact dimensions of the helix, the distance between the bases, and the proof that the phosphate backbone was on the outside. Today we recognize her as a pioneer of X-ray crystallography. Her work did not just solve DNA, it laid the foundation for how we understand viruses and other complex molecules. So, my dear students, whenever you are asked about the discovery of DNA, modern mark schemes often look for an understanding of scientific integrity. Remember, it was a multidisciplinary effort. DNA discovery was a multidisciplinary effort. Chemistry gave us the base ratios, that work was by Shargaf. Okay, physics gave us the diffraction data, that was made by Franklin and Wilkins. Okay, and biology and modeling gave us the structural assembly, and that work was carried out by Watson and Crick. So the lesson for your exam is, and for life, is that science is rarely a eureka moment by one person. It is a slow collaborative process of building on the data collected by others. To wrap up very quickly about our deep dive into the master blueprint, let's look at the DNA discovery timeline. For your revision, do not just memorize the years, focus on how each discovery acted as a building block for the next. In 1950, Shagaf gave us the chemical rules, adenine with thymine and cytosine with guanine. By 1952, Rosalind Franklin provided the physical evidence with photo 51, proving the helical shape. And in 1953, Watson and Crick pulled it all together into the 3D double helix model, which we study today. This journey culminated in 1962 Nobel Prize, cementing DNA as the master blueprint of life. Now, before we move on, here is your big 5 prime summary checklist. If you are sitting for your paper, for your exam, these 5 points are your must knows. Okay, you must know about these 5 points. The elements. Unlike, first of all, the elements. Unlike carbohydrates, DNA has nitrogen and phosphorus. So remember C, H, O, N, and P. The subunit. The monomer is the nucleotide. And always mention the Shargaf rule. Okay, what is the Shargaf rule? Your response, I am waiting your response in the comment section. Okay, what is the Shargaf rule? And second one is complementary base pairing, adenine with thymine and cytosine with guanine. And the method is X-ray crystallography, which was the camera that saw the shape, and the first shape is the double helix, which is twisted antiparallel ladder. If you master this and you have mastered the foundation, then you have mastered the foundation of modern biology. Here, just in this slide, I want to just make a correction if it is not clear, that here is a double bond between adenine and thymine, but here is a triple bond between cytosine and guanine. Okay, so you should not be confused about it. Okay, my dear students, here we conclude our deep dive into the structure and function of DNA. I hope this lecture helped you visualize exactly how life's blueprint operates and gave you a clear understanding of the groundbreaking discoveries that lead us here. If any part of the theory is still a bit unclear, do not hesitate. Drop your questions in the comment section below and I will get back to you for sure. If you found this helpful, make sure to hit the subscribe button and turn on the bell icon so you do not miss any upcoming lessons in our IGCSE biology series. Tomorrow, we will be exploring the next topic in this journey. Until then, study hard, stay curious, and I will see you in the next lecture. Goodbye.